Hello and good afternoon, good evening, or good morning, depending on where you're joining us from. Welcome to this Twist Bioscience webinar on end-to-end sequencing solutions. My name is Chris Thorne and I'm Senior Manager for Field Marketing here at Twist. So before we begin, I'd like to just go through a few items of housekeeping. We will be taking questions after the presentation, and so if you have a question, please submit that in the Ask a Question box. You can move around your screen and amend the sizes of the various pop-up windows that you have and get the screen however you like it. And within those windows, you'll find a related content section where you can find some great related resources. And if you'd like to speak to a member of our sales team, you can click the Contact Us button and a member of the team will reach out to you soon. Finally, when we come to the end of the presentation, if you have 1 minute to take a brief survey, we'd really appreciate your feedback. So thank you for that. And so I'd like to introduce our speakers today. First of all, Alan Fox is a field application scientist at Twist Bioscience, focusing on custom NGS project solutions and new product development. While working in research, clinical, pharmaceutical, and biotech environments, Alan has gained over 15 years of experience in next-generation sequencing, genomics, and molecular biology. He earned his master's degree in biotechnology from the University of Pennsylvania, and he obtained his ACSP certification while supervising and managing clinical molecular genetics laboratories at the Hospital of the University of Pennsylvania and Thomas & Jefferson University Hospital. Our second speaker is Dr. Andrea O'Hara, and she is Strategic Technical Specialist at Azenta Life Sciences and has over 13 years of experience in next-generation sequencing. She earned her PhD from the University of North Carolina at Chapel Hill in genetics and molecular biology and did her postdoctoral training at the National Institutes of Health. So, Alan, over to you.
Thank you, Chris, for the nice introduction. Today, I'm going to talk to you about how Twist can make a customized NGS panel just for you. So let's get to it. Here at Twist, what we do is we make DNA and really good high quality DNA as well. So we take the tried and true phosphoramidite chemistry that's normally done and typically done in a 96 well or 384 well plate, such that you get 96 oligos or 384 oligos. And we twistify the whole process using the power of microfluidics and a silicone wafer. We can get about approximately 1 million oligos per wafer. So you can see how we have such higher throughput. And in doing so, then we're able to help save you time, money, as well as give you still really good quality oligos as well. And this feeds into a lot of our lines of business. Whereas we have a lot of offerings, a lot of ways that we can help out the world, essentially. But a lot of times people come to us about like a SynBio option, essentially making a gene or making an oligo pool or making like a site saturation library or putting anything into whatever you put into a vector or a plasmid. We work with you to help make that construct and help you put it in, and however you want to do. We can do cloning, things on those lines. But realistically, today, I'm really going to talk to you about our NGS offerings, not so much that, but if you have any questions, feel free to ask us at the end and we can get in touch with the right players. And with NGS, what we offer you is an ability to do everything from the library prep to the target enrichment, such that we can provide you a really great enzymatic fragmentation kit that really has low chimera rates, really good ligation efficiency, works really well in FFPE samples, obviously genomic DNA as well. Whatever type of sample you're putting in there, it works really well. I highly recommend it. Or let's say you have a Cavaris or a Sonicator, or you're working with cell-free DNA. We do also offer a mechanical fragmentation kit as well. And then we also offer multiple different types of flavors of adapters. So we have our full-length combinatorial dual index adapters, such that if you're going to try to do like PCR-free whole genome sequencing, I recommend going that route. But if you wanted, let's say, have a little bit more higher throughput, but most of the time people are working with our universal dual index adapters, such that you're putting on a short stubby adapter and then using PCR to add the sample barcode as well as the P5, P7 sequencing components to like an Illumina sequencer. And we have right now currently 384 combinations of that. But let's say you want to do like duplex sequencing or want to go to really low depths of allele frequency, then we also offer a UMI adapter as well, so unique molecular index adapter, to be able to label each strand of the DNA with a molecular index, and then you just use our 384 combinations of PCR primers in that with that adapter. You just swap out the adapter essentially. So we have an option for you, whatever you're trying to work with in that sense of things. And then we also, since we wanted to make sure we're providing you everything that you can do to do the library prep, we also provide you the purification beads, the DNA purification beads, as well as a HiFi PCR polymerase that is really good, high quality, that's just as good, if not better than Kappa's HiFi. And then after you do the library prep, then you would go into our target enrichment steps. In this case, we offer 2 different flavors of target enrichment. It depends on kind of what your workflow is and how you want to go about doing things. If you want to do the library prep and then set up the standard HIBE, which is pretty much a 16-hour overnight HIBE, you can do that and then do the enrichment steps the next day. Or let's say you wanted to squeeze everything into one day and do the library prep as well as the hybridization, then we also offer a fast hybridization solution as well. We obviously offer some fixed panels, which I'll go into way later, but realistically, what we're here to talk to you about is a custom panel. What do you want? We can make it for you and then help provide you everything you need to do that. And we also provide beads as well as the blockers for the hybridization, as well as the PCR enzyme to do the post-capture PCR. So we help you along this way, and we're helped, to guide you anything else that you need as well. But let's get to our custom panel designs and how we go about actually really things you need to take into account, how we go about doing things, and just give you a better idea of, whenever you're trying to come talk to us about your custom panel, what you need to know. First things first, well, there's many ways to go about doing a target enrichment panel. So first thing though is, what genome are you working with? Are you looking to study dog or canines? Are you looking to study monkeys? Are you looking to study bacteria or a certain virus or multiple different strains of viruses or bacteria or fungi? Whatever it is, just let us know the reference genome, and you can even provide us a FASTA sequence as well if need be too. But most of the time and most of the inquiries that we get are human related as well. So, and in that case scenario, we just need to know, well, what genome build are you working with? HD19, HD38, the new T2T genome build, whatever it is, happy to work with you and make it work, of course. But then this funnels into then a bunch of the questions of, well, what are you trying to do with your panel? Are you looking to do diagnostic, like looking more towards hereditary or cancer? Are you looking at mainly hotspots? Are you taking into account some MSI regions? Are you looking at translocations? Are you looking at fusions? Are you looking for any copy number variations? Is there any pseudogenes we need to take into account? We designed so many different types of like cell-free DNA and RNA even panels. We've done so many cancer hotspot panels. We've kind of know it all and have taken it all into account. So we'll give you some advice if it needs it, but you can just provide us the information, and we're very good at this point designing the panel against it. Or let's say you just want to design a panel that's just against genes, whatever your application is that you're trying to do. Well, in that case and scenario, well, if you're just looking for a gene, well, give us the gene name, but then do you want the UTRs? Do you want the promoter region? Are you looking at just the exons? Do you want some tiling in the introns for copy number? Is there multiple different transcripts we should be taking into account, like some pseudo exons, things like that? We have, we just need to know a little bit of information to kind of help making sure that we're making the panel just for you. Or you can give us the coordinates as well. So here's your chromosome start and stop. Or what are you taking into account? Are you doing MSIs? You're looking for STRs, looking at a SNP? Are you looking at something that's more complex, like an HLA gene or a TERT promoter? All this comes into play as well when we're trying to design. So just let us know what you want. And we've done so many different designs and iterations that we're very happy to translate that to what you're actually trying to work with, all that expertise. And one of the other biggest things that also comes into play as well is, well, what are you designing? What do you want us to design against? Are you looking at genomic DNA, or are you possibly looking at RNA as well, or cDNA in this case, because you have to make the RNA cDNA to be able to do any library prep in that sense. But if you're looking at genomic DNA, well, then that's easy. We design the probes accordingly. But if you're really going to take into RNA, we got to really think about, well, if you want to do one probe for both, then we have to take that into account. So if you're looking at, let's say, an exon size of greater than 120 base pairs, like in this example, 240 base pair sequence, our probes are 120 base pairs in length. So in this scenario, we can fit 2 probes snugly right against each other to span that exon. This will work for both RNA and DNA, both these probes, because it just covers just the exon. However, if you have an exon size that's less than 120 base pairs, let's say in this example, 50 base pair exon, Well, if we design 120 base pair probe, we're clearly going into the intronic sequence here to kind of design the probe. So this probe won't work at all, really well, if at all, for RNA sequence, because you're going to need to know the exon to exon boundaries then. So in this scenario, if you want a probe that's going to be working for both genomic DNA and RNA, for this scenario, we need to know the RNA transcript so we can design against the cDNA or, RNA, the exon to exon boundaries. So it's something to come into play every time that we're designing probes. And so we need to know, well, are you working with genomic DNA? Are you working with cell-free DNA? Are you working with RNA or cell-free RNA? All this comes into play when we're designing. So we just need to work with you at what sample type. Another thing that kind of comes up as well a lot is this idea of shadow coverage and how we design our probes. So like in that example previously that I showed you with 240 base pair exon, with us then putting 220 base pair probes in there nice and snugly, not going into the intron. We don't like to pad into the intron if we can avoid it. We like to make sure our probes fit snugly in the exonic regions. And in that case and scenario, then if you care about splice site mutations, well, if you're sequencing 100x coverage of the exonic region that you're looking for, you still, through the power of shadow coverage, just because DNA is randomly fragmented, you're still going to be sequencing a little bit extra on the ends of what you're trying to pull down. In that scenario, you'll get about approximately 80x coverage when you look about 30 base pairs out. So that is really still just as good for what you're trying to do with splice site, mutations, things on those lines. People are very happy to know that they didn't have to waste more sequencing of that intronic region by us padding into it. And you still get, some other coverage as well further out when you're looking at 50 or 100. You're still going to get some lower coverage of those just because it's a bell curve. And that's just due to the random fragmentation and our probes being able to pull out, for bigger pieces of DNA, things along those lines. That being said, if you ever want any padding into it and you want us to take that into account, just let us know, and we're happy to design our probes accordingly. One other biggest thing too about with our probes is we kind of understand how they work really well. We've done so many iterations of panels and things like that, if you, let's say you want to design against a wild type sequence, well, could you still pick up the mutated sequence? And of course you can, your probe will work, but to some extent. And this diagram is trying to show you is, if you had a continuous, let's say, mismatches across the sequence. So let's say you had 10 base pairs or 20 or 30 base pair mismatches, continuous sequence. So whatever reason, it's completely different. Then you still have, let's just say, a 20 base pair continuous mismatch error. We still have 100 base pairs of the 120 base pair probe. So the 100 base pairs is still well enough to be able to pull up this sequence and be able to enrich for it as well. And then you'll just sequence that 20 extra base pairs that mismatches against it, through sequencing as well. But let's say you have randomly dispersed mutations across that 120 base pair probe region that you're trying to tile. Well, DNA stretches, bubbles, all that stuff to try to work. But at some point, it's going to, the stoichiometry and the chemistry is going to break down. So this happened, when you look at essentially after 10, 15 base pair mismatches, it can start breaking down and then it won't really work as well. And in that scenario, then we might want to design a new probe. And this comes into a lot of times when, this comes into account a lot of times when we're trying to think about like viral strains or bacterial strains or things along those lines, when you're trying to look at many different viruses or many different bacteria and try to use least amount of probes as possible and condense it down to, our probe mismatch tolerance will be able to capture all these strains if you go up to 10 base pairs or 5 base pairs mismatch, things along those lines, to then help save you money on, having less probes, things along those lines. So we take this into account every time that we're designing a panel. And speaking of SNPs as well, most of the time people just only want a SNP. They only want one SNP when they're looking at a panel. So in that case, most of the time we really just try to do 120 base pair probe spanning the SNP region that you're asking for right in the middle. However, we do have a repeat filter that we take into account a lot of the repetitive regions of the genome, like ALUs, lines, and signs, and can adjust accordingly the probe distance away from the SNP, or at least overlay, based on these regions. And this always comes into play when we're doing a design, and something that kind of makes sure that we're giving you an optimized probe that will work for what you're trying to do. And if you ever want to design against a SNP, all you really need to do is provide us the chromosome, start and stop. And a lot of times, though, we get a lot of inquiries about copy number variation and whether or not our probes would work for it. Well, the good news is, yes, of course it will. And we can have our tried and true strategy if you're just looking like our exonic or one of our exomes, or you're just trying to say, all right, I want all the exons of a gene. I still want to look at hotspots, things like that. So we do our standard method of doing the tiling spanning across the exonic regions. And then however, though, this will work just fine. But let's say you only really care about copy number. You don't care about mutation hotspots or anything like that. Well, we could just put probes, sparsely across the exons such that then that will help still give you copy number as well. And still translate to what you need. So realistically, whatever the option you go with, whether you're just looking at normal mutation calling and then having the probes still be used for copy number or spanning every exon, or you just want a few exons in there just only because it's for copy number, we can still make it work for you. And all you need to do in that scenario is provide us the gene or the region and desired resolution as well for this copy number, and we'll help design that panel for you. And a lot of times we get a lot of requests for, well, indels, insertion deletions. There's things that go in, that come up as well, especially like FLT3 ITDs, things like that. So we do have design strategies for that. First things first is we usually design a flanking region against the probes. And this also comes into account for like MSIs or STRs as well, where we have designed flanking probes against it and then use shadow coverage to kind of fill in the gaps. But if you know the sequence in the middle, then we can also design a center probe as well. So in this case, we're looking at a wild type sequence where we have, the normal, sequence in there for that probe, and then the two flanking ones. Well, when the mutant happens, this part is deleted and comes out such that this probe that centers the actual deletion might not work. And then you're just relying then on the two flanking probes to, pretty much get the sequence that you want. And then conversely, for an insertion, same concept, two flanking probes with a probe right over the region, the wild type region that you expect an insertion. Though while this insertion, since the probe is kind of right in the center, it might not, it still might fail and might not work as well. And then in this case, in scenario, you're going to rely on shadow coverage, like I was mentioning, like MSIs and STRs or any variable region. Then you'll rely on the shadow coverage to kind of fill in the gaps, and the two flanking sequences will pick it up. And then, but if you had a really large insertion, let's say about 100 base pairs or whatever it is, we can design a probe specifically over top of that region and be able to then just pull out that large insertion, give us the FASTA sequence, give us whatever it is. We can design a probe right over that and still have the two flanking probes as well to help capture it if need be, knowing that it could be a variable insertion. And in this case and scenario, you usually just need to provide us the 5' and the 3' coordinates of each of the, spanning the region of interest indel, and the indel length. Do you expect it to be 10 base pairs? Do you expect it to be 100 base pairs? Whatever it is, just let us know. We'll help do the design for you. Most of the time, this is one of the biggest questions I get is about fusions, and many different ways of doing it. Are you looking at it at the DNA level or the RNA level? In this example, I'm going to kind of give you for the RNA side of things, but different, we have different strategies for both, whether you're doing DNA or RNA. So in this case, just two fused genes coming along together. So what you can provide us is the combined sequence, whereas you can say, here's the coordinates and the directionality, and then we can combine them together and we'll make a hybrid fusion for us to then design the probes against. Or you can also just provide us the FASTA sequence. So here's the sequence, here's the fusion transcript ID, whatever it is, so that we can find the fusion event to help design the probes against. And then what next thing that we do, we can extend about 240 base pairs out from the break point, from the fusion point, and that will then help us design extra probes if need be. So then we can either use this extension and design like two probes here on the either side of the fusion to be able to pick it up, and then rely on shadow coverage if there's any variation. Or if it's a known fusion, it's BCR-ABL, it's EML4 ALK, those, the exact fused sequences, we can design a mutant probe right over top of it, or we can, do a combination of both. However it works, whatever you need, we can help design. And we have really good expertise in a lot of fusions already in our database with probes designed against. And then kind of taking a different turn to kind of think about designing is going to a different concept is long read capture. So we just announced a partnership with PacBio, and we can help work with you to help design a capture panel to help enrich for your long read regions of interest, like 3 KB, 5 KB, 10 KB, whatever the size, whatever it is, we can help work with you. And in this case scenario, we have learned that you don't want to do our conventional tiling strategy because you have too many probes on these longer fragments. They're biotinylated. I failed to mention that earlier. All our probes are biotinylated. So that biotin, if you have too many probes, can pull and break down then your DNA fragment, such that we have to do some sparse tiling across it to then help pull out and enrich for the regions to help keep it more intact. So happy to work with you. We have a lot of customers working on this right now. And another thing that we also do as well is methylation. So we have a kind of a complex probe designed to really help enrich for methylated regions as well. And we work with NEB with their EMC kit to kind of do the first part with the library prep, because it wasn't on my that one slide that I mentioned. But we have a library prep for you option that's really gentle on the DNA using enzymes to help do the conversion. But then we can help you with the probes and doing the enrichment for your methylated regions of interest. And how we go about doing this is we actually design 2 sets of probes, one for the unmethylated and one for if it's fully methylated region. Then we design probes against the antisense and the sense strand, such that we have about 8 probes in that one region of interest that you're looking for to be able to then capture the DMRs, everything, the methylation status, everything in between, just relying on our probe mismatch tolerance that I mentioned earlier. And going through all this, giving you the ideas with the design and all that stuff, that's the very first part. So usually you just reach out to us, we reach out to you, however you get in touch with us. The design process is completely free. All you have to do is submit all those interests, like I mentioned to you, the chromosome start and stop, what gene, whatever you're trying to look for, what you're trying to do. We do the design, provide you the design in a day or two. Here's the file, here's what we designed against. You can look at it in UCSC, you can look at it in IGV. Let us know how it looks, how it goes. You're ready to hit go. You generate a PO, all that stuff. Then we build it. It takes about 3 weeks for us to build the panel, much faster than a lot of our competition. And then you can then test it. And then if there's anything that you have to adjust accordingly, maybe some balance off, whatever it is, we can go back through this whole process again in the same time that usually you would get a panel from one of our competitors. And I'll admit, very little time do we go back and redesign. Very rarely does that happen. Very few scenarios does that occur. And after you make your panel, one of the other things to take into account too is I talked to you about making a custom panel, but another thing that you can do is make a custom spike into one of our already fixed panels. So let's say you like our comprehensive exome or our exome V2, which is our industry leading spec exome. It's really great quality, really great on target, really great low duplicate rate, things on those lines. But let's say you want to spike anything into one of our exomes. Well, we can help work with you on that as well, make a custom spike into a boost or adjust certain regions. Or let's say our comprehensive viral panel, which has like 3,000 viruses across it, including like monkeypox and SARS, if you want to use for surveillance, and you want to add different types of viruses like other mammalian or other like bird viruses, whatever it is, you can also add a spike in for that. Our methylome, which is our big gigantic methylation panel, that is about on par with like doing more than an epic array essentially. You can also spike in different content with there, or even our pan-cancer methylation panel as well, which is more cancer specific. It's a smaller focus panel spanning a lot of different cancers and CBGs in that sense of things, or even our canine exome. So I mentioned that, we can make a viral panel, we can make a bacteria panel, we can also spike into our canine or even our mouse exome as well if need be. So in these scenarios, let's say you like our fixed panel, but you want to add to it. So we can add whatever it is, whatever the content is. We have a CNV panel. We have a SNP panel, diversity SNP panel for human. We can add mitochondrial. We can add whatever custom spike in you want. We can help make a custom panel for you to spike it in, or we can blend it together into one panel and provide that just for you. So up to you. We're here to work with you and customize however you need be. And here's just kind of the list of your sales reps, your local sales reps in this area that covers New York down to DC area. Feel free to reach out to them if you have any other questions. And thank you so much for listening. If you have any other questions for me, please put them in the Q&A box and we'll get to them at the end of the presentations. Thank you very much.
Thanks, Alan. Okay, we're going to hand over to Andrea now.
Hello, my name is Andrea O'Hara, and today I'll be presenting Azenta's end-to-end sequencing solutions using Twist targeted panels. So first, a brief introduction about Azenta. You may know us by our previous name, GENEWIZ. Last fall, we rebranded under the Azenta name. This actually includes the union of a number of different companies, including Brooks Life Sciences, GENEWIZ, as well as many others. With this union, we actually offer a number of different products and services really spanning the entire sample exploration and management. So this includes genomics and analytical services, which we will be discussing some more today, sample sourcing, storage, automation and logistics, consumables and instruments, and data and informatics. And all of these come with consultative services. Now, as I said, today we're specifically focusing on this genomics and analytical services. We have a number of different sites located across the globe. Our next generation sequencing headquarters are in South Plainfield, New Jersey, as well as facilities in Leipzig, Germany, and Suzhou, China. As you can see here, though, we actually have a number of different facilities really across the globe here. So depending upon your project needs, you actually have a number of different options with regards to where you can submit samples for later processing. Now, today we'll be discussing our next generation sequencing. We have a number of different next generation sequencing options, but today we're really going to focus on this targeted sequencing section here. And so when we think about the next generation sequencing options, Azenta offers complete sample to analysis solution, meaning that we're going to start all the way with the experimental design and take this through to the final project delivery. Let's dive into this just a little bit deeper so we can discuss what all these different steps entail. So the first part is going to actually start out with your experimental design. Now, depending upon what you've been working on with your Twist representatives, you might have already discussed this in larger detail, but depending upon where your project is and what your project needs are, you might be considering something on the whole exome line, which would allow you to use a predetermined kit to determine variants for germline, or if you're looking at oncology samples, regular somatic mutations or low frequency mutation detection. This is really ideal if you're not quite sure yet what your targeted areas are going to be. You have a roadmap towards that targeted panel, but you are trying to decide which ones you want to pick to start with. You might already have some targets in mind. So you may have already designed your Twist targeted panel. And again, this is going to be a custom design where we're going to go for an increased sequencing depth, and we'll be able to identify a number of different things, not just SNPs and indels, but also other variants. Now, depending upon where you've already picked this, you might have already discussed this. If you haven't already discussed it and you're thinking about a targeted panel, we also work very closely with Twist for these designs, so we can also help bring this union together. But when you're discussing that approach, you'll also get to discuss everything else that's going to be included with this study design. And this will be with a PhD level manager, might even be somebody such as myself, that will be able to actually discuss this in more detail. We can provide tech notes and case studies, go over that full sample to analysis solution. Now, once you're ready to proceed, you would start out with submitting your samples. Of course, you can always submit the purified genomic DNA, but you can also submit a number of other samples depending upon what you might have. So this can include things like cell pellets, fresh frozen tissue, FFPE, blood, many other types of samples. If you have a particular sample type that you're interested in, you can always feel free to reach out, or you can go to our sample submission guidelines page and actually look to see what types of samples are accepted and how much you need. And of course, if you have a really custom sample, let us know. Really, there's very few limits to the options here. We can accept most sample types. Now, once that sample has been received and the DNA has been extracted from it as needed, the next step is going to be sample QC. All samples will undergo a sample QC to make sure that we have plenty of DNA and that it's of a high quality. The next step is going to be library preparation, and in the United States, all processing is based in US for US projects, meaning that if you submit your samples to New Jersey, they're going to stay in New Jersey. It's highly automated and optimized, and this really allows us to get great reproducibility. It also allows us to scale up so we can do both standard size projects as well as very large projects, and it reduces the costs. Now, again, we are thinking here about either the whole exome or a custom designed targeted panel, and that library preparation process will happen with this automated equipment. And because we have all this automated equipment and as little hands-on as possible, that really allows us to, again, get that reproducibility and reliability and also reduce any possible risks. Now, once the library preparation is complete, we're going to do QC on the library. This is to make sure that our libraries look good, we're getting the expected yield and library size. Next up is sequencing. For Twist panels, we're typically going to be using Illumina short-read sequencing. However, we do have a number of different sequencing solutions available. That includes PacBio, 10x Genomics, as well as NanoString. But again, here for this one, we're typically going to be looking at Illumina sequencers. And with this one, we have a suite of different pieces of equipment here, ranging from that smaller MiSeq to the mid-range HiSeq, and then the large-scale NovaSeq. And we'll help you discuss and pick which one's going to be the best option based off of the size of your panel, number of samples, and your desired sequencing depth. Now, once the sequencing is complete, we're going to do another round of QC, and that's really just to make sure that you're getting all of the data that you expect. So we want to make sure that you're getting both the yield you're anticipating as well as the quality of data. Next up will be data analysis. You'll always receive your raw data regardless of what option you pick, but we do have the option for you to add on bioinformatics analysis, and this includes both preset analytics packages as well as customized bioinformatics, depending upon what you need. This is going to include HTML reports as well as a number of different files, and the contents of them will really vary depending upon what kind of analysis you need. The last step here is going to be project delivery. Typically, we're going to be delivering through SFTP. If you have your own cloud set up in AWS, perhaps we can deliver directly to that. But we also do offer the old-fashioned version of just sending you the hard drive. So depending upon whichever option you need, we're happy to deliver in that fashion. So this is really taking us through this entire end-to-end protocol for that targeted panel. But what about the next steps? So if we think of the central dogma where we have DNA going into RNA, going into protein, we've covered our DNA here. We're really going to learn so much more about whatever it is that we're studying, the underpinnings, mechanisms of the disease, things that might be impacting treatment options, any number of different details. But we've done that targeted panel here to really learn more about all of the samples that we're looking at. The next thing that we want to think about is that RNA expression or transcriptomics, really taking this to the next level to say, I know I have these DNA modifications. How is that affecting what's being expressed in the RNA? Now, when we're characterizing samples via transcriptomics, we have a number of different products and solutions that we can use for this. This is really showing us a high-level view of all the different options we have available. So first, we have our Illumina-based RNA sequencing, a few different methods here: poly-A selection, rRNA depletion, small RNA. This is our most traditional bulk sequencing option. Next up, we have PacBio. This is long-read technology, and this is going to allow us to look for full isoform sequencing or identify alternative splicing. Depending upon the goal of our targeted sequencing analysis, we may opt for an isoseq approach, especially if we're looking for novel isoforms that are maybe due to a particular a variant that we've detected, or if we're looking again for those alternatively spliced variants. 10x Genomics is single-cell RNA sequencing. This is going to allow us to look at the expression profile within specific cells, which will allow us to say when we're looking at particular tissue types, as an example, really identifying how things might vary within any particular cell type. Again, important things to consider if we're thinking about a more complex tissue that might have a number of different cell types present that might be impacted differently with whatever those results were from our targeted sequencing. And then last up here, we have the NanoString GeoMx digital spatial profiling. And this is going to be true spatial analysis, where we'll actually be taking those tissue specimens, looking at a slice through it, and looking within that 3D space to learn more about the transcriptomics expression, but within the context of the tissue itself. Now, again, though, our most traditional approach here is typically going to be that Illumina-based RNA sequencing. And so if we look at the RNA sequencing in more detail, I'm showing you here a high-level view that's really talking about how these different platforms vary. You see I have boxed out here in purple, mRNA sequencing and total RNA sequencing. mRNA sequencing is really going to look at just that poly A tail. So this is all of the RNA that's going to be turned into a protein. This is our most traditional approach, and this is what we're going to be using to determine what's being expressed. It's a totally unbiased approach, meaning that we're looking at anything that has the poly A tail, and we're going to be able to see what's being expressed at a high level and at a low level, and we're going to learn more about that general expression profile for the samples that we're evaluating. We also have total RNA sequencing. We're still going to be focusing our analysis primarily on that poly A tail, but we're actually going to start with a pool of all the RNAs, which means that we'll also get to see things like long non-coding RNAs. Especially if we're concerned with any sort of regulatory aspects, we can really start to get a little bit more insight when we're looking at that total RNA versus just the poly A. The other thing to consider here is our sample type. Most of the time we're going to be working with fresh samples, but if we have something, say, FFPE based, we may opt for that total RNA sequencing, which is just more amenable to some of the degradation that we might get when we've fixed the samples. Now you'll see here I have a few different options also shown. So again, we have a high throughput option if you're looking at a large number of samples. Again, depending upon our study, we might have 96 to 384 samples, in which case that might be a good alternative. And then again, I'm also showing that IsoSeq approach. If the variants that we're interested in that we've detected are going to have an impact on transcripts that we might have novel transcripts or do discovery on, that's where we might want to consider that option here. But again, most traditional approach here is going to be doing that bulk RNA sequencing option. Now, regardless of which option you pick, we still have bioinformatics solutions here as well. So our general bioinformatics solutions are going to be based off of our gene expression. We'll get some slice variants and gene ontology analysis. We also can provide mapping and hit counts. We can look at SNP and indels, gene fusions. We can even do some de novo assembly or detect those novel isoforms. So again, a lot of different options here depending upon what you're interested in. And just like with our targeted sequencing approach here, when I talked about that full workflow, you'll get that same consultation with this. Other things to consider is that when you're talking about us with about that primary project, you can actually start discussing if you want to do RNA sequencing at that time, because we do have the option, depending upon your sample type and interest, of doing both RNA and DNA extraction from the same sample, which means that we would be able to really look at that DNA and the RNA from within the same sample to get that really thorough context of what's happening. So we're not even looking at different versions of something similar, but in fact, the exact same thing. Now, when we're thinking about this central dogma, again, we're going to talk about those DNA changes and then the RNA telling us what's being expressed. But really, at the end of the day, protein is king. Protein is what is going to be actually doing all of that heavy lifting here, and that's going to be our end result. So I like to think of the protein analysis or proteomics as being our final frontier. And we actually offer some really excellent proteomic solutions. So for protein biomarker discovery, we use Olink. Olink is really ideal for learning all sorts of different types of proteomics questions, and these can be very specific targeted or some more general broad discovery-based things. Olink as a technology is really excellent for proteomics because it's highly sensitive. It has a wide range of acceptable sample types and small sample volumes, as low as 1 microliter. And in addition to all of this sensitivity and variable sample types, it's highly specific. And this has to do with its dual antibody method using their PEA technology. So the way that the PEA technology works, there's a little picture here shown on the slide. If we have the protein, we have 2 antibodies, dual recognition antibodies for any particular protein. If you guys are at all, if you've done proteomics before, you'll be aware of the fact that one of the hardest parts is really just getting specificity for your antibodies and getting removing non-specific binding. So using this dual recognition antibody, we're going to have 2 different antibodies that will recognize the protein of interest. If I just got one, that's not enough. I need to have them both. And they need to be the right match. So we're going to have these dual recognition antibodies, and you can see it has this sort of whisker barcoded. If it's a perfect match, meaning that I have, this is protein A and I have antibody A1 and antibody A2, and they're the correct match, these dual linked barcodes will actually link together. And that will offer us that really great specificity that we need. So we're really reducing the potential for cross-reactivity. Again, very critical because we're now not looking at one protein, we're going to be looking at many, many proteins. Very sensitive. This is, if you're, again, familiar with proteomics, this is similar or even more sensitive to ELISA. That dynamic range spans greater than 10 logs, and it's a really high throughput. We can look up to hundreds or even thousands of proteins. Now, with regards to our sample type, we want to think of basically any sort of biological material here, or biofluids, as I like to think of it. So we're going to be looking at something that's in a liquid format, but this can be things like tissue lysates, cell lysates. It can be cell culture media, also human fluids, so saliva, tears, plasma, blood, any number of different products. If you have a particular sample type that you're interested in, again, discuss with us, because more than likely there's going to be either a direct or an indirect biofluid that we'll be able to use for this Olink technology. With Olink, everything is actually going to be processed within the CLIA-CAP lab, but it's for both RUO and clinical samples, and it has a fast turnaround time. Now, one of the biggest selling points here for Olink, though, is the fact that we can look at not just one protein, but many, many, many proteins at once across many, many samples. So we have a number of different targeted panels to choose from here. There's the Target 48, which is the standard panel for cytokines. Target 96, which has a number of different panel options, depending upon the disease or information that you're looking for. And then the Explore 3072. Now, the Explore is really going to allow us to do that very broad proteomics approach. So again, depending upon where our research is and what we're looking at with regards to that, additional primary targeted panel, any sort of transcriptomics that we did, we may decide which of these different assays we want to use for that downstream proteomics analysis. We're happy to provide you the information for the targets for these, so you can actually know more about what the different targeted proteins are to help you choose what might be the best assay for your analysis. And again, in theory, depending upon how you have this all set up, you can actually use that exact same sample to do your targeted panel, RNA sequencing, and proteomics, all from the same sample. So you're really just going to get that most robust, very thorough result. Now, the overall workflow, similar to what we saw earlier, you're going to start out with your experimental design, submit the samples for processing, and then you're going to get the downstream data analysis. This actually comes with data analysis automatically included, and it's going to come with both a certificate of analysis as well as a quality control report. And within that, you'll also get all of the normalized protein expression values. So here you're going to really get a lot of really excellent quality control information included with that analysis, just to ensure that you're getting that appropriate sensitivity and specificity that we're looking for here with the proteomics assays. Now, if we look at our different platforms here, when we're thinking about that targeted DNA sequencing, this is where we're really focusing specifically on identifying those particular mutants, variants, SNPs, indels, whatever they may be. Again, we can think of a broad targeted panel like whole exome sequencing using the Twist mouse or human panel, or we can do a custom Twist design panel based off of whatever targets you have of interest. I didn't touch on it today, but we also do offer the Twist methyl-seq approach. So if you're thinking about looking more on the regulatory side, also an option here. Next up, we have the bulk RNA sequencing, and this is again going to be taking us from that to that next step. So starting with that DNA, but learning what's actually being expressed and how does that alter expression based off of what we would anticipate in a normal sample. And then finally, we're going to be looking at that protein biomarker discovery using Olink. And this is really going to allow us to see what sort of protein level changes are occurring. And again, depending upon that targeted panel we've picked upstream, we might really be able to see a lot more detail about how that's actually changing or looking with that final downstream protein expression. Now, we know that you have a lot of different partners that you might choose to work with when you're thinking about your study. So I'd like to just give you a little bit of points here to consider about why you might want to choose Azenta. So as I mentioned here, we really focus specifically on that targeted sequencing with some of our other approaches, but we have a whole suite of options. So depending upon what you're looking at, really a lot of things to consider. We are one of the highest on-time delivery rates for next generation sequencing, and we have a ton of experience. So over 20 years of experience over a wide range of sample types and a wide range of species that we've covered. We have a comprehensive online portal, excuse me, which allows you to order everything online. You'll be able to track everything online. You can share your results with colleagues as needed. And like I said, you can even watch as your status progresses through our online portal, along with a dedicated study manager for your contacts. Now, in addition, we do have vigorous QC metrics that we perform with every single sample and every single study. So we already discussed about that high-level sample QC. There will be a library QC, post-sequencing QC, and then if you have analytics, analytics-based QCs. And we're really strive for excellence here. So we actually have above industry standard measurements for all of our next generation sequencing. And again, you'll have a dedicated person in the US to contact if you ever have any questions or need any support. Now, finally, when we actually think about that quality of data, like I said, we do have one of the highest on-time delivery rates within the industry. We have expedited options available. So depending upon what you're looking for, if you're looking for something that's going to be the most budget friendly or the fastest possible, we have whatever option to suit your needs. And again, all of this coming with that complimentary consultation to discuss further and help you choose your best option. And so with that, I'd like to thank you all for joining us, and we are happy to take any questions. Thank you.
Okay, thank you, Alan and Andrea, for a great presentation. Very much appreciated. So now we're going to open it up to questions. We've had a few come in already during the presentation, but if you do have questions for either Alan or Andrea, or both, then feel free to put those in the Q&A box and we'll try to work through them. So maybe we'll just begin with a question for Alan. Perhaps, Alan, you could begin by telling us, how does Twist handle novel fusion designs? So you mentioned the fusions during your presentation, but how would you approach novel fusion designs?
Yeah, no, it's a great question, actually, and not something I went too much into detail. So one thing that we go about doing is realistically just add more probes. So we throw more probes at it. You can go up to 4, 6, 10 times. We can add as many probes as necessary, tiling the regions that we know the known breakpoints. So if you know where the breakpoint approximately is, we can just add more and more probes there to be able to then capture through like shadow coverage and be able to hopefully capture that chimera read in that sense of way of doing it.
Okay. And similar question perhaps from Tapan, who asks, for CNV, who I'm looking for unknowns. So sounds challenging, but how would Tapan go about designing probes in that context?
Yeah, so that's another great question. And obviously, if we don't know the sequence, it kind of does make it challenging to figure out where to begin. But if you have an idea of the chromosome, we can tile across the chromosome in that sense of things, like by doing about 1 probe every 1 megabase. We can do it up to many different tiling in that sense. We can go up to like 1 kb or 1 in every 10 kb or 1 every 100 kb to really get in the hone in that region that you're looking for. We actually here at Twist have 2 panels that kind of behind the scenes, but we have an option to do like a copy number panel where we do have 1 tiled across the whole genome, 1 every 1 megabase, so it's about 3,000 probes. And then we even go to a really high resolution and go down to like 1 in every 100 kb, a lot higher amount of probes, but there's options there where we can just tile across the genome or the chromosome or the region of interest, however the spacing that you're trying to, and resolution that you're trying to look for, and go about it that way.
Yeah, and I think that's something you were getting at in one of your slides, wasn't it, whereby with Twist you can kind of go through that iterative approach. So if you don't get it quite perfect or you learn something on the first pass, then we can go through multiple iterations to get you to your ideal probes.
Yeah.
Perfect.
Exactly.
All right, well, I've got.
Go ahead.
Sorry. No, I was going to switch over to some questions from Andrea, actually. So, Andrea, in your presentation, you talked about the three parts of the workflow: the targeted sequencing with Twist, the RNA-seq, and the Olink, of course, as well. So, for the proteomics. So, is it possible to use the same tissue sample for all three assays?
Yeah, that's a great question. So yes, you actually can use the same tissue sample for all three of these assays. Now, if you're interested in doing that, that's something you're going to want to mention in advance. So you can do a dual RNA and DNA extraction. It can be from the exact same cells, or you can partition the sample and do one DNA and one RNA. You also can use your tissue lysate. You can take just a little bit of that tissue lysate out and use that for the proteomics. So if you set this up sort of for your best success, you know that you want to get this trifecta of options from the get-go, you can actually use that exact same sample to do all three assays. This would be true for tissue samples as well as for any sort of cellular material. So if you just had like cells in suspension, you'd also be able to do that with those.
Perfect. So presumably you can, if someone chooses to work with you, you would provide some guidance on the sort of minimal sample amounts, but provided they show up with enough material, then they're good to go on all three assays.
Absolutely. And so, again, one of those things to consider, even if you already have your panel already designed and ready to go, that early project consultation phase that I discussed, that would be a time where we could discuss what are the different layers you're looking at, how much would you need to be able to provide to do that. And honestly, one of the nice perks of this is that you could choose to do these one after the other, but you could actually do all three simultaneously. So you can run your Twist panel at the same time that you do your RNA sequencing and Olink, and you would get basically all of those results back at the exact same time. So you can kind of have it all running at the same time, so you get all of this data at the end of the project.
Yeah. And on a related note, so in terms of what organisms you can do this with, so would this workflow be suitable for zebrafish, for example?
Sure. So actually, Alan, if you'd like to go ahead and answer that question regarding the targeted sequencing using, any other organisms.
Yeah, no, thank you. Sure, yeah, I'll jump in. So Twist can design any panel against any provided reference genome or even FASTA file. So we get a lot of requests, not so much about zebrafish, but a lot of viral, bacterial, monkey, canine, cat, dog. I've seen it all so far in that sense. So as long as you give us what reference genome to use or just give us the sequence, then we're able to help design a panel, any targeted panel.
And then regarding RNA sequencing, exactly the same concept here. As long as it has a reference genome here, we can do RNA sequencing and get you those results back with full analysis. Olink currently is only available for human and mouse, though. So that's going to be the one caveat because these antibodies are going to be species specific. So for the proteomics, we're going to be really thinking only human and mouse, but for the RNA sequencing and targeted sequencing, any organism that, again, as long as we have that reference genome, we're good to go.
Perfect. So, Alan, question here about the Twist pan-respiratory viral enrichment panel. So, around the panel itself, are you able to go into a little bit of detail about how the panel is built? So, are they tiled probes across the genome or random sparse probes? And do Twist only include probes against known human respiratory virus sequences or all sequences available in the NTBI for viral families? So, I guess within the limits of what you can say, what is, could you provide a little bit more detail about the viral panel?
Yeah. I'll, as quick as succinct as I can. That was a lot of questions. So each one of the viruses on at least the respiratory viral panel is 4x tiled. So there's a lot of probes going against each one of those viruses that are on there. Our other panels, like our SARS one, is also 4x tiled. It's really, more geared towards SARS, but SARS-CoV-2 is still in the respiratory panel as well. So it's influenza, RSV, all the good respiratory viruses and that sense of things. We have a lot of people using it too, getting really good results. they're designed against and like you're mentioning, the NCBI sequences that are out there. And, we tiled it in a way to ensure to be able to capture as many of the viral strains as possible, especially influenza, to really be able to pick up any diversity that is there. So there's a lot more that goes into the design process, and, we're happy to, share any more information later, but we can also, always add more diversity if it's need be as well. Because I know for RSV, it only targets like A, B, and C of the virus strain. Sometimes there's more that people want. So there's more that we could always do and spike in and add to it if need be.
Perfect. Well, we are coming up on almost a full hour, 55 minutes. So I think in the interest of everyone's time, we'll sort of wrap up there. I do want to thank our speakers again for presentations, for two fantastic presentations, and remind our attendees that if they wouldn't mind, there'll be a short survey right after. Andrea, just quickly though, before we do go, if people do have additional questions about Azenta, what's the best way for them to reach you guys?
That's a great question. So if you do have any additional questions, you can feel free to reach out to us by email at NGS, Next Generation Sequencing, NGS@azenta.com.
Perfect. And similarly, if you want to find us, you can reach us at twistbioscience.com and contact us through our Contact Us page. And we look forward to having some great conversations in the future. So thank you, everyone who was joining us today. Thank you again to our speakers, and we'll see you at the next webinar. Bye for now.